let's start chapter 3 in Bio 105. Now, chapter 3 deals with the cell, its structures, and its functions. Now, a lot of these structures we covered in lab, so I'm not going to double hit us on those structures. But I do want to talk about other structures that we have that are not on the lab modules. Okay, so the first is cytoplasm. So cytoplasm is this jelly-like fluid that suspends all the cellular structures. So, you know, we have the plasma membrane all the way around, nucleus, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, mitochondria, centrioles, lysosomes, ribosomes. These are all stuff we talked about in lab. So this right here is where the cytoplasm is. It's in between those things. All right. Next are peroxisomes. So this is showing a peroxisome. So peroxisomes are vesicles that contain enzymes that break down poisons. Now these poisons are like alcohols and peroxides, and that's where the name derives from, peroxisome. So we produce alcohols and peroxides as a byproduct of our metabolism. But we don't want those to develop up to toxic levels in our cells, so we have these peroxisomes which absorb those poisons to break them down. Next is a cytoskeleton. So you can see on here there's all these little fine fibers in there. That's part of the cytoskeleton. These are filaments and tubules that provide support for the cell. If we go to this next picture, you can see those a little bit more there. So that's the cytoskeleton. Now, we do have appendages. So appendages are structures that stick off of our cells. So the first are flagella. So this is showing a flagellum here. Flagellum are long whip-like appendages. And what they do is they move the cell. And the only cells that these are found on are on sperm cells when we're talking about humans. They're found in other organisms, but with humans, only the sperm cells have a flagellum. Next is the cilia. So here's the cilia down here. These are shorter, more numerous, hair-like appendages. Now, what they do in us is they move substances. So this is showing a top view of cilia here, and this is showing a side view of cilia. And so cilia line our respiratory tract. So they line our nasal cavity, our sinuses, our trachea, which is our windpipe. And so why they're there is in those areas they're going to move mucus. So why do we produce mucus then? Is because whenever we inhale air, we don't just inhale air, we inhale anything dissolved in that air. Dust, dirt, pollen, bacteria, viruses, whatever's in that air. And so the mucus that we produce is to trap that stuff. And then through ciliary action, we're going to move it to our pharynx. So if it's on our sinuses, we're going to drain that mucus down into our nasal cavity. And from our nasal cavity, we drain it to our pharynx, our throat, and then, I just swallowed some mucus. If it's down in our bronchioles or bronchi or trachea, here ciliary action is going to move that mucus up to our pharynx or our throat, and then, I just swallowed some mucus again. Every time we swallow some mucus that is drained by ciliary action from our upper respiratory tract down to our pharynx, or from our lower respiratory tract up to our pharynx. Now I do want to point out, smoking inhibits ciliary action, and so smokers, and you actually stop producing cilia. So smokers, the only way to get that cilia out from their lung area is to cough it up. And you know, this is the least of your worries though. That's not pleasant, but smokers are at increased risk of asthma, emphysema, and 80% of all lung cancers are associated with smoking. Anyway, oh yeah, and if you live in Madison County, we have the second worst air in relationship to airborne particulates that cause cancer. Yay! Anyway, let's move on to the plasma membrane. Okay, so the plasma membrane is the boundary of the cell. All right, so it is a selectively permeable membrane. It is made of a phospholipid bilayer that's embedded with proteins, and it's going to regulate what enters and exits the cell. And this is a blow-up of the plasma membrane. So, as I said, phospholipid bilayer. So this is one layer of phospholipids, there's a second layer of phospholipids. And all these structures that we see here are proteins. All right? Now, obviously, our plasma membrane is not blue. Proteins aren't all these various different colors. They're fairly colorless. All right? So let's take a look at these phospholipids. So So, I know this might be hard to see, but I'm just going to go ahead and draw one of these phospholipids. So here is a phospholipid. Okay, so a phospholipid has a phosphate head and it has two fatty acid tails on it. Now that phosphate head is said to be hydrophilic. So what hydrophilic means is that it's water loving. So these are molecules that are attracted to water. And these two other parts, these fatty acid tails, they're said to be what are known as hydrophobic. So literally water fearing. So these are molecules that are repelled by water. All right? So when we take phospholipids and we put them into water, what's going to happen here is these phosphate heads are going to orient towards the water, and the fatty acid tails are going to move away from water and towards each other. And so that's what we see here on this membrane. So the fatty acid tails have moved towards each other, the phosphate heads have moved away from, have oriented towards the water. And so this is what makes the plasma membrane this selectively permeable membrane. So we have this large layer right there in the middle that is hydrophobic, water fearing. Water can't even move through there, all right? And so what that does is it makes this great interface between outside of a cell and inside the cell, where this can be a very different solution than what's found inside the cell. So the only things that can move directly through that phospholipid bilayer are things that are also hydrophobic, okay? And so this is really, really good in a lot of ways because, you know, if you took a shower, let's say within the past 72 hours, your phospholipid bilayers, your plasma membranes did not dissolve away because water cannot go through those. So really, really nice because of that. Okay, so only small and hydrophobic molecules can. Now, let's take a look at other lipids that are found in the plasma membrane. Another one are these little guys here. These are cholesterol. So cholesterol helps stabilize the plasma membrane. All right, so because the plasma, these phospholipids are kind of watery, so it's going to help hold them together. Next in here are glycolipids. So this is showing a glycolipid, well, this is showing a glycolipid right there. So glycolipid, this is a phospholipid with a carbohydrate attached. So you can see one there, there's another one there. These guys are used in cell recognition, all right? So I'm going to talk about those with the next ones, and those are glycoproteins. Glycoproteins are proteins with carbohydrates attached. They're also used in cell recognition. So we see one here, there's another one there. All right, and so what do I mean by used in cell recognition? Well, we have these cells in us, our defensive cells. Those are white blood cells. And they go around our body and they try to find things that don't belong to us. And if they don't belong to us, they try to kill them. All right, so that's what these white blood cells do. So they bump into cells, and if those cells have different glycolipids and glycoproteins, different identification tags on them, they're going to kill them. All right, if you have the right identification tags, those white blood cells leave you alone. All right, that's essentially how our immune system works. All right, so these are identification tags, glycolipids and glycoproteins. So this is one of the problems when we talk about organ transplants because, you know, if you get somebody else's organ, your white blood cells are going to try to kill it. All right, so that's why we have to give you immunosuppressing drugs when that situation arises. Okay, so blood types are types of glycoproteins. So if you have A blood, you produce the A glycoprotein. If you have B blood, you produce the B glycoprotein. If you have AB blood, you produce both the A and the B glycoproteins. If you have O blood, you don't produce this glycoprotein. So one of the things about O blood is it's a universal donor. Everybody can accept O blood. And the reason is that if something lacks identification tags altogether, our body doesn't understand that it could be harmful, or it, you know, treats it as it's harmless. But this is nice in that we can put metals and plastics into us. So if you had a tooth filling or hip replacement, you know, your body's typically doesn't reject that stuff unless you have an allergy.